Let me quickly revise what is isoelectric point. We use this term isoelectric point to express that a molecule mainly exists in a Zwitter ion form, and it will not exist in this form or this form. That means the quantity of only cationic form or the anionic form would be quite less. If majority of the component of the amino acid only exist as Zwitter ion, then the pH at which it is existing is called as isoelectric point. Clear? How do we find that isoelectric point? We try to figure out both ways. If this one, the Zwitter ion, is accepting H plus, then this is the cationic form. If it is giving H plus, this is the anionic form. From the cationic form to the Zwitter ion form, the equilibrium constant existing, let us call it as Ka1. From Zwitter ion towards the anion, the equilibrium constant that is existing, let us call it as Ka2. This is the dissociation constant for acid. So think this as an acid. In the forward direction, K1 is the dissociation constant. Here, K2 is the dissociation constant for this reaction. Okay. And now, isoelectric point by formula will come out to be pK1 plus pK2 by 2. So basically, it's an average of this K1 and K2 value. Now, how you need to understand that? Check it out. For example, if at all I take any amino acid, generic amino acid as in like this, I have, you know, NH2 here, COOH here, H here, and R here. And now imagine if this R group is something like hydrogen or methyl, isopropyl, isobutyl, any of this. Basically simple alkyl. If you have simple alkyl, that means I have one COOH group and one NH2 group. Then what do we call it? We call them as neutral amino acids. Correct? So if you have only alkyl group, we call it as neutral amino acids. Generally, for all neutral amino acids, if you recall, we learned about 6 neutral amino acids. Gavli, remember that? Glycine, alanine, valine, isoleucine, leucine, and isoleucine, and phenylalanine. All these 6 are simple neutral amino acids. For neutral amino acids, when you add their pK values and calculate, the pi value turns out to be 6. The isoelectric point will be very close to 6. That means it will be somewhere close to 5.5 also, maybe 6.5 also. On an average, it will be 6. Understood? While if by chance, if that amino acid in this R group also has something like OH, SH, or amide, CONH2. These groups are also present in it. Okay, like cysteine. Okay, those kind of groups are present on it. Then the isoelectric point comes out to be 5.5 on an average. That means it could be somewhere around 5 or 6.5, say up to 6 also, but on an average around 5.5. But what you would notice, if there is an extra COOH group here, then this molecule will become acidic amino acid, correct? Because you will have 2 COH compared to that of 1 NH2. So it is an acidic amino acid. For acidic amino acid, the isoelectric point will come out to be close to 2 to 3. Okay. And now, if you have an extra NH2 group in this alkyl group here, there is one NH2 group here. So then there are 2 amines versus 1 acid. So this will become basic amino acid. So for basic amino acid, the pi value range anywhere from 8 to 10. Histidine is, a basic amino acid slightly, with an aromatic ring form on it. That's why it is not exactly 8. So the exact value for histidine is 7.6. Okay. So broad way, if you think out of 20 amino acids, everything will fall in this everything will fall in these four categories only. This one is an exception. Got it? Clear, beta? Now, just for the sake of understanding, notice if there is a neutral amino acid, can you notice this is a neutral amino acid? There is one COH versus one NH2. Then it is neutral amino acid. So for neutral amino acid, the pK values are given. So what do? You simply take the average of the pK values. That will be your PI value. That is isoelectric point. Okay. Second, if at all we have two acids and one amines, then take the average of only acids. That will become your PI value. Clear, no? And if you have, you know, two ammonias, and one acid, two amine groups and one acid group, then take the pK values of only amines. So PI is 11. Okay. But notice this case. Here we have three COH groups. If you have more than two COH groups, take the average of the pK values which are at the least values. For example, 1 and 2. Take the average of those two. That would turn out to be the PI value. Similarly, if you have more number of ammonia or amine groups here, more than 2, then take the pK values of the highest values. In the case of acids, you will take the lowest values. And in the case of amines, you will take the highest values. Got it? So that makes it PI value as 11. Clear? I hope this point is clear to you guys. This is how you calculate the isoelectric point. Got it? Where is the application of isoelectric point? To know that, first we need to understand what is peptide bonds. This is also which I discussed with you. If two amines are combining with each other, they will combine by dehydrating the water and converting into an amide bond. You can see this side there is some amount of amine. This is another amine. Amino acid. Between these two, the connection got established by removing the OH group from acid and hydrogen from NH2 here, from here and C double bond O. So if I remove this water molecule from here, you would establish peptide bonds. So this amide linkage is what we call it as peptide bond. Got it? For example, if I take alanine and glycine, alanine structure is like this, glycine structure is like this. I need to put this NH2 close towards COH here so that I can remove that out. So H from here, OH from here, remove it, you will get an amide bond. And make sure while you are writing something like this, alanine and glycine, then acid and amine will combine. They will form an amide bond. Keep this NH2 on the left hand side and keep this acid on the right hand side. Okay. This is a convention that need to be followed. Always keep nitrogen on the left hand side and the acid part on the right hand side. The nitrogen side is called as N terminal. The carbon side is called as C terminal. Okay. Similarly, if you notice, alanine and glycine, the way you combine is different than glycine and alanine. Now notice glycine and alanine combination. So basically OH from here, H from here, you are removing and you make this compound. Now if you clearly notice between these two, they are structural isomers, but they are not the same compound. Because you can see the N substitution here is different. N substitution here is different. Alkyl group here is different. Alkyl group here is different. Okay. Hence, a combination of, a combination of these two could be different if you change the alignment as well. Okay, so remember that two amino acids can never be same when they are combining with each other at different combinations. Which brings to the fact that if there are two amino acids combining, we call them as dipeptide. If there are three, tripeptide, then tetrapeptide. If it is 10, it is decapeptide. And how many number of peptide bonds you will get? Two amino acids will make one bond, one peptide bond. So which means if we have 10 number of amino acids, we should have 9 number of peptide bonds. Every time there is a bond, there is a removal of water molecule. That's why same number of peptide bonds and number of water molecules lost will be same. Okay. Now, if you have two different amino acids like A and G, okay, so number of dipeptides possible will be equal to A and A and G and G and G. So how many possible number of dipeptides? 4. Similarly, if I want to make tripeptides out of using only two of them, then you can notice AAA, AAG, AGA, GGA, GGA, and these are all the peptides which you will get. Have you noticed the pattern here? So we can see 2 raised to power of 3. Those are the number of tripeptides we have. So here this power factor basically represents how many number type of peptides you want. And here this number represents the number of amino acids that you are taking. Correct? So if n number of amino acids you take and y are the number of types of peptide linkages you have, then can I say that n into y plus 1 is the number of possible peptides, because y is the number of peptide linkages. So number of amino acids which are present will be, I mean number of type of peptide that will be possibly forming will be y plus 1. Okay, so therefore n raised to power of y plus 1. Clear? Any doubt there? Right. For example, I have AGL and I want to make dipeptides. There are three of them and I need to make dipeptides. Okay, for dipeptides, that means you will have only two. So power should be 2. And number of amino acids are 3. So 3 raised to power of 2, 9. So can you see these are all the dipeptides. Similarly, I have A, G and L and I want to make tripeptides. How many possibilities? 3Q. That makes it 27. Simple maths. I'm sure you guys understand that part. Correct. This was something which was asked in previous year question, that if you have a decapeptide and on complete hydrolysis, glycine, alanine and phenylalanine are separating out on hydrolysis. Now glycine alone contributes 47% to the total mass. Then I need to find out how many number of glycine units were present in decapeptide. You know that 796 is the decapeptide molecular mass given. But when you hydrolyze that, again you are refilling all the peptide bonds with water molecules. For a decapeptides, how many peptide linkages we will have? 9 linkages. And on 9 linkages, how much everywhere you are adding what water molecule? So this is the total mass of alanine, phenylalanine and glycine together. Correct? That is the total mass. Out of that total mass, 47% is a contribution from glycine alone. So you can see 47 by 100 into 958. So this much is the contribution alone coming from glycine. So which means 75, which is the molar mass, into X, which is the number of glycine molecules. That unit should be equal to 450. So X should be equal to 6. Clear? Right. Notice this other question. This was a very recent JE question. Okay. To solve this, I told you that first you need to understand how the molecules behave, you know, when amino acid is kept in a electrolytic cell, okay, at different pH values. This is a glycine molecule, CH2COH and NH2, glycine. Glycine, you know, its existence is always this way. This is the zwitterion form, correct? This is the zwitterion form which it exists. Now, this zwitterion does not move to cathode, it does not move to anode as long as it exists mainly in the zwitterionic form only. Got it? That means when will it stay there? When the pH of the solution is same as its pi value. I told you that the pi value of neutral amino acid ranges close to 6. Correct? This is the isoelectric point. So you can see if I take cathode and anode, if I take cathode and anode at pH is equal to 6, at pH is equal to 6, okay? So this would become CH2COH and NH3 plus, okay? Which means, sorry, at pH is equal to 6, it will exist as this way. This is the zwitterionic form, it will exist. Which means it is neither going to go towards cathode, neither it is going to go towards anode. So there is no movement. This X means there is no movement. And the sign, overall sign is 0 because plus and minus makes it 0. There is no movement, moving towards cathode or anode. But when you keep pH is equal to 1, imagine karo, I took that same amino acid and kept in this solution where the pH this time is equal to 1. And if the pH is equal to 1, this being a neutral form, it will try to take H plus and convert into this form, which means now what is the charge on it? Positive charge. So it has become a cation. Cation will always move towards cathode. So therefore the sign is positive and the movement is towards cathode. Got it? Similarly, if I keep pH is equal to 10, then this compound, which is a neutral compound, when you keep the pH as 10, it feels that this, because this isoelectric point is 6, it feels that now the solution which you have added is basic. And since it is basic, what will it do? It will give away H plus and it will convert into this ion. And what is this ion now? Anion. This is the anion. And anion will move towards anode. Clear, girls? So when pH is equal to 10, the movement will be towards anode. pH is equal to 12 means even better, more basic. That means that will also go towards anode. Got it? Have you understood how the movement of ion is? Both of you, right? Now, check out the same thing in a question where this is the structure given. This is the structure given. And there are two different R positions given, R1 and R2. And you notice that its isoelectric point will keep varying based on R group. Now, if both the R's are simple alkyl groups, okay, if both the R's are simple alkyl groups, what would happen, beta? If they are simple alkyl groups, definitely there will be pi maintained at 6. Correct? Pi will be maintained at 6. Similarly, if this R1 and R2, any one of them is changed, and one of them has become acid or an amide or something, then pi will accordingly change. Now notice first case. If you have both being hydrogens, this is a hydrogen and this is also hydrogen. If both of them being hydrogen, will there be any movement? No, no movement, because it will exist as a zwitter ion. Even if you have a hydrogen and methyl, again it will remain neutral amino acid, and neutral amino acid does not move around pH is equal to 7 or 6, because the isoelectric point for neutral amino acid is very close to 6 and 7 only. Correct? 5, 6, 7, honestly, you will not see much movement. Okay? 5, 6, 7 is like the averaging of all the neutral amino acids. Next, you see there is an acid here and hydrogen. So definitely now there is an acidic amino acid. And what is the pi value of acidic amino acid? It is around 2 to 3. And since it is 2 to 3, now the pH 7 feels like basic solution to it. And since it's a basic solution, what will it do? It will give away H plus. And when it gives away H plus, charge will be minus. So you can see it will become minus and start moving towards cathode. The other side, I have an amide and an amine. If you have an amine, the pi value will be around 9, 10. And for 9, 10 pi value, that means isoelectric point, pH 7 looks like acidic. And since it is acidic, the lone pair from nitrogen will take H plus from the solution and becomes a positive charge. And this entire thing will become positive charge. And since it becomes positive charge, where does it move? It will move towards anode. So it will be positive and moves towards anode. If this is amide and amide, so pi value remains close to 6 and 7 only. So no movement. Both of them are amines. The pi value will be even higher. So it will be somewhere around 11. So 11 is even more basic. So if you put that in pH 7, that compound will believe that the solution is acidic, and it will again try to take H plus. So it will again become positive charge. If one is acid, one is amide, because there is an acid part, definitely it will again have the pi value close to 2 to 3. And if it is 2 to 3, 7 becomes basic. It will give away H plus, gets a negative charge. If again there is amine and one more alcohol, uh, if you have OH, SH, double bond, amide, these are all not going to make any difference. Okay. Hmm. But you can see one side there is an amine. So definitely again it will become, pi will be higher value. Pi will be around 9 to 10, something like that. So for that pi value, pH 7 appears like a acidic solution. So it will take H plus from the solution and gets positive charge. Last one is also same case. So the question asked was, how many of these peptides will be positively charged at 7? So answer is 1, 2, 3, 4. So that was the answer. Girls, have you understood this previous year question? Clear, right? On a similar lines, this was one more question just recently asked in PYQ. The structure of the peptide is given this way, and you were asked what is the absolute value of net charge if pH is 2, pH is 6, pH is 11. Okay. See, when it is neutral amino acid, you notice that here we have, one acid, acid, amine, amine. That means equal number of amines, equal number of acids. So that makes it neutral amino acid. So since it is neutral amino acid, the pi value will be somewhere around 5 to 6, correct? Clear? So if we have pH is equal to 6, do you think there will be any movement beta? No, that means it will act like an isoelectric point only. So when pH is equal to 6, will there be overall charge observed on this one? No, overall charge will be 0. So that's why Z2 is 0. Second, when pH is equal to 2, of course, this pi value being 5, 6 feels that now the solution is acidic. Since it is acidic, it will start taking H plus. So where all H plus it can take? It can take H plus here, it can take H plus here at two locations. So if we take H plus at two locations, correct? What do we get? If it's plus two locations, the total charge will be plus 2. So that's why Z1 is 2. Agreed? Last, when pH is equal to 11, this was very interesting one. pH is equal to 11. Of course, if the pH is 5 to 6, it will feel that the solution is basic. So it will start giving away H plus. From which all position it can give? It can give H plus from here, it can give H plus from here. And we are forgetting the fact that phenolic is also acidic at pH is equal to 11. Agreed? So therefore, phenol will also give away H plus. So how many H plus are we getting out? 3. So we will have minus charge on at 3 locations. So magnitude of the charge is 3. So sum of Z1, Z1, Z2, Z2, Z2 and Z3 is how much? 5. Else, is this clear?
Yes,
This was what we learned. Now let's talk about what is the structure of protein. Protein can be classified based on its structure, right? Which is again 4 parts. One is primary structure. Primary structure refers to the entire sequence of amino acid. Okay. For example, glycine, alanine, valine, phenylalanine. Okay. This is a sequence. Okay. If the sequence is differed, it will result in a new protein. Even one position, at least at one position, if you change anything, that means it is a new protein altogether. Got it? Next. Okay. If you remember insulin is a protein. Okay. And insulin will have 51 amino acids. Got it? In a perfect sequence. And that is the exact sequence that need to be followed. Then only it is named as insulin. Okay. Secondary sequence refers to the configuration in which the polypeptide chain exists. Okay. That means if you have alpha helix or beta plated sheet, these are the two configurations. What do you mean by that? See, for example, if you notice from here, this particular line is the sequence. What is that sequence? This is the primary sequence. Okay. In the primary sequence, if you notice, clear, there will be hydrogen bonding existing from here to here like this. So because of the hydrogen bonding, right, instead of staying straight chain, it will start coiling around. And that coiling appears like a helix. That's why it is called alpha helix. Got it? So protein chain will coils into right-handed helical structure, and this is done because of its intramolecular hydrogen bonding. Similar to that, if you take multiple protein chains, this is within the protein chain, right? You might also have like this. There is an N terminal here, C terminal here. So this is the complete protein sequence. Now, between these two sequence molecules, somewhere hydrogen bonding can establish here like this. So if the hydrogen bonding is being established between the sequential chains, between two chains, okay, and usually they are anti-parallel to each other, correct? So then that is called beta plated sheets. Clear? Again, this is also formed because of the intermolecular hydrogen bonding. Have you noticed here? This is intramolecular, this is intermolecular. And next, 3-degree structure. 3-degree structure refers to the further folding of the 2-degree structure. So the 2-degree structure which you have, either it is in this form or it is in this form, beta plated sheets. If that will start further coiling around, okay, again it will give you two forms. One is called fibrous, the other one is called globular. Okay, right? So that means if I take a small part of this, that itself is a helical part. Understood now? This much part is that helical part, and next one is another helical part. Between that helixes again, there is a strong bonding established. Clear? Then that makes it fibrous form. If the winding is not regular and it is randomly going everywhere, that is called globular form. Okay, clear? Usually our muscles majorly have, you know, fibrous form. That's why muscles are longer, elongated. Got it? They're all proteins only. Got it? So you can take silk, keratin. Keratin is the protein which is there in our hair. Wool, myosin, right? These are all different forms of fibrous proteins. Globular. Globular means hemoglobin in our body, insulin, albumin, egg whites. Albumin is egg whites. Correct? So that is all globular form. Understood? So tertiary will be again categorized into two forms, fibrous and globular form. Okay? You can see the forces which stabilizes 2 degree and 3 degree, right? 2 degree and 3 degree can be formed by multiple types of attractions. What are those? It could be hydrogen bonding. It could be having disulfide linkage. What does that mean? See, if you notice insulin, right? Insulin will have a chain like this of 21 uh, amino acids. And then there is one more chain of 30 amino acids. Between these two, there is a linkage of sulfur and sulfur. That's how it is connected. That's how it becomes insulin. Okay. So the two longer or primary sequences are being connected by sulfur-sulfur linkages. Agreed? So that's how 2 degree, 3 degree can show hydrogen bonding, disulfide linkages, or basic van der Waals attractions. These are the types of forces of attraction you will notice. Next one is 4 degree structure, tertiary structure of protein. Tertiary proteins will have two or more than two polypeptide units. Two or more than two polypeptide units called as subunits. Okay. It is like this. This is one globular form. This is one globular form, and globular form. So it is a 3 degree structure. But they four together form one unit. This is typically how hemoglobin also happens to happen. It is like this. Iron is somewhere at the center, and there is one sequence of proteins here, one sequence of proteins here, one here, like this. Within that, we'll have multiple sequences here, making it as tertiary, and they are all connected to iron like this. So that makes it, hemoglobin. Got it? So if your tertiary units are converting into small subunits, then that is called quaternary structure of protein. Okay. So I hope you guys understood. There are four types of structure of protein: primary, secondary, tertiary, and quaternary. See, proteins are generally biologically active, and when they are active, they are called native proteins. That means there is a life existing in it. But when it is subjected to heat or chemical treatment, it might lose that biological activity, and that is called as denaturation. The process is called denaturation process. For example, boiling of egg, correct? So there is a biological activity which was going on, which we have stopped by heating it. Clear? So that is called denaturation. So during this process, 2 degree or 3 degree structures are destroyed, but there will not be any change in the 1 degree structure. That means the primary sequence of the protein does not change. It will remain the same. Only the secondary, tertiary, and quaternary get destroyed. Okay. Any doubt in this case? Okay. See, the particles which are present in the nucleus of a cell, that is responsible for our heredity. Okay. And this is what we call it as chromosomes as well. Okay. And in fact, chromosomes are made up of proteins and various other biomolecules. And out of that, one of them is nucleic acid. There are two types of nucleic acid within the chromosomes, which is DNA and RNA. DNA is deoxyribose nucleic acid. RNA is simply ribonucleic acid. Okay. Nucleic acids are a very long chain polymers of something called nucleotides. And if you make it as a polynucleotide, which means one molecule is a nucleotide, okay, one particular molecule. If I make it into a big chain, that will become polynucleotides. So how does it happen to have? See, if you take a complete hydrolysis of DNA, right? If you take a DNA and try to hydrolyze that, you will get one pentose sugar, that is ribose. We'll get ribose. Then we will get a phosphoric acid, H3PO4, and a nitrogen containing heterocyclic compound. A heterocyclic compound which is nitrogen containing, and these are called as bases. In short, DNA is made up of three main components: sugar, base, and phosphoric acid. Okay, not just DNA, RNA also. Okay, and DNA specifically is made up of sugar moiety. I mean, the sugar moiety, that means the sugar part which is present in DNA is beta D2 deoxyribose. Okay, just a second. When it comes to DNA and RNA, you will see there is one important difference. Both of them will have same sugar ribose, but in DNA, at the second position, you will have deoxyribose, which means there is no OH group. This is the actual structure of beta D ribose. But at the second position, if you notice, there is supposed to be OH in a ribose group. But if that OH is been removed, that compound is called as deoxyribose. Clear? So this sugar moiety which you have, this the difference between RNA and DNA is mainly this. The second important difference is DNA majorly consists of four different types of bases. One is adenine, guanine, cytosine, and thymine. While RNA contains four bases in which the first three are same, A, G, C are same. The last one, thymine, which is for DNA, while RNA will have uracil. RNA will have uracil. Now notice, adenine and guanine have very similar type of structure, correct? So it is two 5-membered and 6-membered rings connected with each other, you know, fused with each other this way. Got it? You can see there is an alternative nitrogens. Okay, here alternative nitrogens and here alternative nitrogens here. And only difference is here we have one NH2 group, while here we have NH2 and OH group. That makes it adenine and guanine. While cytosine, thymine, and uracil will have similar group. Again, you can see alternative nitrogens here also, like this. Basically, it is the same structure. This ring is same in all of them. So make sure that that part is clear to you. So this ring is same in all of them. Except here you can see in cytosine we have C double bond O. And here we'll have, you know, instead of NH2 here we'll have C double bond O. And here also we'll have C double bond O and C double bond O. Correct? And without this methyl group. So thymine and uracil will have that difference that there is a methyl group here. Got it? These are the 5 bases which you need to know. Now, a unit formed by attaching the base with the sugar is called nucleoside. Remember the meaning of it. Side and tide. Okay, so this will be nucleoside. So If the numbering of sugar is made as 1 cross 2, 2 bar, or sorry, 3 prime, 4 prime, 5 bar prime, like this can be distinguished with a base having 1, 2, 3, 4. Okay. And when the nucleoside is linked with phosphorus at fifth position, say at fifth position, now we'll make a phosphor ester. Okay. So this is becoming, the side has become tied. Understood now? The nucleoside has become nucleotide. Okay. So in total, you can see this is the structure we have: a sugar, a base, and a phosphoric acid. Okay. How do we make the nucleotide into polynucleotide? To make it into a polynucleotide, now from the third position, again you make an ester, and that same phosphorus you connect that to fifth position of the next sugar. And that's how it will keep on going. Third and fifth. Again from here, you will have a connection. Again from here, you will have a connection like this. Can you notice that now? So that's how a polymer form can be done. So once the phosphorus, which is monoester, has converted into phosphodiester linkage, that means it has started forming polynucleotide. If you have only two sugars here, that means it is dinucleotide. If you have three, trinucleotide. If you have n number, it is polynucleotide. Got it? And just like in protein chains, we call it as a primary sequence. Here also, the first sequence of the polynucleotide chain is what we call it as primary structure. Okay, got it? Now, RNA and DNA has this one more difference. Have you noticed? So far, we found two differences. One is with sugar, one is with the type of bases connected, and the third one is the type of structure. While when it comes to RNA, there is only single strand. Only this one strand is there, means that is RNA. But when it comes to DNA, it has been found that it will have two strands. Okay. So one strand which is going from here, this is a polyphosphate formation at fifth and third position getting connected. Similarly, from the other end, we have a 5' and going all the way towards 3' connected. Okay. So anti-parallel. So when they have anti-parallel two sequences, then they will start showing hydrogen bonding between them, between the bases. Okay. And here is the interesting part, what kind of hydrogen bonds it is making. See, if you notice, in the DNA, we learned that AG, AG, CT are the bases which are used. Now, between G and C, there will be three hydrogen bondings forming. And between A and T, you will have two hydrogen bondings forming. Got it? That triple line which is given, it does not mean a bond. It's not a chemical bond, it is a hydrogen bond. Okay. So it will start making this intermolecular hydrogen bonding, which is 3 between G and C and 2 between A and T. And that's how the sequence keeps moving. And this type of double helical sequence is what we call it as DNA. In RNA, usually we will only have one sequence like this. Got it? So the same sequence, sometimes if it is very big, it will go like this and wind up again. But it is not a different sequence in RNA. But in DNA, we'll always have two different sequences. Is this logic clear? Okay, right. RNA is again made up of three types of RNAs depending upon functionalities. There are three types of RNAs: messenger RNA, ribosomal RNA, and transfer RNA. Okay. Again, the details of this is what biology students will learn. Okay, there is a separate chapter for them, so we will not have to get into that detail. As much as DNA and RNA is concerned, we need to just learn what is the basic structure, how does the nucleoside, nucleotide, polynucleotides are forming. Clear with this part? The next thing we need to understand is what is photosynthesis. Okay. See, usually whenever you have carbon dioxide, which is released from, sorry, which is absorbed by plants, it will use moisture or water along with sunlight and chlorophyll. Chlorophyll is a magnesium complex to form glucose, correct? Now, ATP is the energy that you are taking, correct? So, 3 ATPs are required to fix 1 mole of carbon dioxide. So, if I want to get 6 CO2 molecules, that means I require 18 ATP. This much amount of energy is actually required by, this much amount of energy that is being utilized by plants to convert that into carbon dioxide. Sorry, this is not carbon dioxide. This is O2. Okay, 6 moles of O2. Clear beta? Now, notice this 6 moles of, glucose plus O2. While we do this now, okay, when we consume, when we have glucose in our body, that will take oxygen, convert that into carbon dioxide, and generate this much amount of energy in our body. How much? 38 ATP. Got it? So when you oxidize one glucose, now I would suggest we'll not have to get into details of it because this is completely bio form of it. You just need to know the numbers, okay? So 38 ATPs are generated when you oxidize 1 mole of glucose. But have you noticed when it comes to 38, the net ATP gain is only 3 because 2 ATP are consumed when, pyruvic acid enters into mitochondria, correct? Like this. This is pyruvic acid, correct? And this pyruvic acid would require, when it has to enter into the mitochondria, it will require another 8 of it. Got it? So which means overall, out of 38 here, correct, the net gain of ATP is only 36. Okay, because here it is again giving away 6 CO2 here. So net amount is only 30 ATP. Okay. So basically 30 ATP is what is built up in our body. That's all we need to understand. Okay. Next thing is about vitamins. See, vitamins, only thing you need to learn is what is the name of the vitamin, what is its deficiency. Got it? So you can see vitamin A, B1, B3, B6, B12, B5, vitamin D, E, K. And what are the deficiencies, what are the diseases which cause, right, if they are deficient. Last thing you need to know is this one. Vitamin D, E, K, these are fat soluble, while vitamin B and C are water soluble. Okay. And vitamin C structure wise is this one, which you call it as ascorbic acid. Got it? It has two chiral centers, if you notice, here and here. These two are the chiral centers it has. Done, beta. Okay. Right. So this is how much you need to know it from biomolecules. Got it?